Hey everyone, it's Hia. Before we start, I have a question for you. We have the power to change the world, and I know you've probably heard this a lot of times before. But have you considered changing the world by reinventing it? Imagine a world where we could reprogram yeast to make medicine and microbes to create biofuels, or create synthetic organisms that can actively monitor their environment for toxins and pollutants, and then execute remedial steps that would involve degrading the pollutants to restore healthier ecosystems worldwide. Using synthetic biology, we can essentially take parts of natural biological systems, characterize, simplify, and convert them into components for redesigned biological systems that can solve some of the world's biggest problems. What sets this field apart from gene editing is that we are not transferring gene from microbe to microbe. Instead, we are creating microbial genomes with new DNA traits from genetic parts, which are then inserted into a cell or microbiome itself. Synthetic biology is basically the convergence between engineering, biology, and computing. If you're new to the area, you can check out the link in the description box below to read my crash course article on the topic, where I cover topics such as programming DNA, DNA sequencing, designer genomes, biobricks, and so much more. The field truly marks a change in the way we think about engineering cells. As synthetic biologists view cells as programmable machines, we can now go beyond utilizing individual genes and begin writing code that does not exist in nature. So in this video, I'm going to be talking about the modeling and computing side of it all. CAD softwares, or in other words, computer-aided design tools, make it possible for scientists and biotech enthusiasts around the world to program or reprogram life right on their computers. CAD enables in silico design, which means to perform experiments or simulations on computers. Whether it be creating, modifying, optimizing, or analyzing designs, it can all be done on these softwares. It's important to note that CAD's features are pretty different from Tinkercad, among other platforms that only use visual interfaces. On the other hand, CAD platforms combine visual interfaces with programming APIs such as Python and Ruby. The complexity of the program varies on the API as well as what you actually choose to build or simulate. A good knowledge of engineering design principles, construction methods, and programming skills are what I would call some of the skill sets you should have beforehand. But this doesn't change the fact that with some learning, anyone can start experimenting with building models as early as today, even if they're really basic. Through learning, we can identify how they can be manipulated and hence unravel all the aspects of complex cell behavior to leverage the unique qualities of different elements of nature. One of the most studied prominent organisms that's considered to be a mainstay wherever microbiology comes into play is E. coli. It plays a pretty critical role in our world today, and its identity goes beyond just a bacterial species that inhibits our digestive tract. It's actually the best understood and studied organism on the entire planet. Its genome sequence was published approximately 25 years ago, which is incredible considering this was one of the earliest sequences to be completed. Characteristics like its versatility, ease of handling, and genetic manipulation, ability to anaerobically grow it at body temperature, along with no specific dietary needs, has made it extremely easy to culture right in labs. There are actually 16,000 genes across the pangenome of E. coli, meaning all of its strains. However, since less than 20% of these genes are present in all strains, it's pretty flexible. What makes this a great organism is that we have the ability to incorporate elements right in the genome. Scientists are growing E. coli in flasks within an incubator, and this process is optimizing the amount of cells produced, so it has become a model organism worldwide for hosting foreign DNA sequences. There are several other applications ranging from metabolic engineering, where E. coli is manipulated to produce desired chemicals during growth, to E. coli-based biosensors detecting environmental hazards or measuring cellular processes. It's clear that E. coli's use in biotechnology is going to persist, and before we can design, build, and test experiments involving the organism or something potentially completely different, we need to learn. We have to understand the building blocks of biological systems and the underlying mechanisms that convey how organisms perform specific functions. I found this almost hidden world of E. coli really fascinating, so this is why I decided to model the lac operon in E. coli using Tinkercell, a CAD software for synthetic biology. It's an open source visual modeling tool with biological parts that have corresponding attributes to define them. Using the parts, models can be constructed and analyzed with Python and C programs through the API. It is one of the older applications, but because of the flexible framework, it can be used despite the changes in the past couple of years in the field. And these changes could be how the parts are characterized, the methods for analyzing the robustness of networks, and so on. So just to give you guys an overview or a refresher for some of you, an operon is a group of genes with similar functions controlled by one single promoter and transcribed as a single mRNA. The amount of an operon transcribed is controlled by regulatory DNA sequences, as they are binding sites for regulatory proteins. It's able to obtain usable energy from a variety of sources, which is another one of the characteristics making it ideal for lab growth. One of these sources is lactose, when glucose is unavailable, hence the name black opera. Understanding how E. coli metabolizes sugars, specifically the disaccharide of lactose, helps us understand at a deeper level about how E. coli's feeding requirements are fulfilled when they're artificially grown in a lab, and how they can potentially be further optimized. It is a heterotrophic organism, so it gets its food from other sources for carbon. And for E. coli, this is glucose molecules ingested by its host organism, and they break it down into usable energy through central metabolism. Now I'm going to give you an overview of the deterministic and stochastic simulations I was able to make of the lac operon using Tinkercell, the CAD platform. Standardized databases of composable models and the Registry of Standard Biological Parts are amazing resources to consider visiting when you're ready to make your own genetic circuits, mathematical model simulations, or anything. I'd also recommend reading Synthetic Biology of Primer, which is a great book throughout this journey. Computer-aided design software development is driving innovation in biotechnology and serving as an opportunity for society to learn about nature at a much deeper level, and for us to model our creative ideas and imagination and to see them come to life right in front of us. I'm really looking forward to continue exploring the space and keep learning about how I can leverage synthetic biology and its many applications to make it.